Welcome, folks. Today, we're diving into the fascinating world of space mutagenesis and its generational impact on rice. We'll be discussing a recent study that explores how spaceflight affects rice genetics across multiple generations.
That sounds incredibly interesting. So why is it important to study how plants respond to spaceflight in the first place?
Well, understanding plant responses to the space environment is crucial for enabling plant cultivation during long-term human space missions. Imagine astronauts needing a sustainable food source. That's where this research comes in. Plus, it might even offer new advantages for terrestrial agriculture. It's a win-win.
I see. Interesting. What are some of the specific challenges that plants face in space?
Spaceflight presents a unique set of abiotic stresses. One major factor is oxidative damage, which affects cellular DNA, tissues, and organs. This damage comes from the direct hit of DNA and water molecules by heavy ions, but also from normal metabolic processes within the plant.
Oxidative damage, got it? And how does this oxidative damage lead to what you called stress memory?
That's a great question. Excessive ROS, or reactive oxygen species, production during spaceflight leads to oxidative stress. This stress can damage DNA beyond repair, causing long-term effects on DNA structure and genomic instability. Think of it as the plant remembering the stress and passing that memory down.
So this study focused on rice. Why rice, and what exactly did the researchers do?
The researchers used a rice mutant called B10, which was identified during the SJ-10 return satellite mission. They assessed the cross-generational genetic effects of spaceflight stress memory by cultivating this mutant over multiple generations, from M1 to M5. They then used a bunch of omics techniques, genomics, transcriptomics, and methylomics, to analyze the changes.
Omics. That sounds complicated. Can you break down some of the key findings for us?
Sure. One of the key findings was that the oxidative stress effects induced by spaceflight persisted all the way to the M5 generation. The rice genome remained unstable, leading to alterations in genome methylation levels. They also saw significant changes in the methylation levels of transposons.
Transposons? What are those and why are they important?
Transposons, also known as jumping genes, are DNA sequences that can change their position within a genome. The study suggests that these transposons are involved in the intergenerational inheritance of spaceflight-induced stress memory. Basically, they're helping to pass down the stress memory.
I see. So the spaceflight is causing changes to the DNA, and those changes are being passed on. What about gene expression? Did they find any differences there?
Absolutely. They identified thousands of differentially expressed genes, or DEGs, and differentially alternatively spliced genes, or DIS genes, across multiple rice generations. Many of these were related to oxidative stress, specifically the generation and scavenging of reactive oxygen species.
Differentially expressed and alternatively spliced. It's a lot to take in. What does it all mean in simpler terms?
Essentially, spaceflight stress is causing certain genes to be more or less active, DEGs, and it's also affecting how those genes are spliced, meaning how the genetic information is put together, DAS. Both of these things are influencing how the rice responds to the stress.
Got it. Now, you mentioned methylation earlier. How does DNA methylation fit into all of this?
DNA methylation is an epigenetic modification. It doesn't change the DNA sequence itself, but it affects how genes are expressed. The study found that genome methylation was associated with gene expression, but not with alternative splicing. So it's another layer of regulation that's being influenced by spaceflight.
So, to recap, spaceflight causes oxidative stress, which leads to DNA damage, changes in methylation, and alterations in gene expression and splicing. And all of this is being passed down through generations. Is that a fair summary?
That's a pretty good summary, yes. The study provides a comprehensive insight into how spaceflight-induced stress is inherited in rice. It uncovers novel mechanisms of oxidative stress-induced genomic instability and epigenetic regulation in plant stress inheritance.
This is fascinating. But what are the practical implications of this research? How can it be used?
Well, by understanding these mechanisms, we can potentially develop strategies to improve plant adaptation to stressful environments, both in space and on Earth. We might be able to breed or genetically engineer plants that are more resilient to spaceflight conditions or even to other stresses like drought or salinity.
That makes sense. So, what were some of the specific methods used to determine these changes across generations?
They used a range of methods. For example, to measure oxidative stress, they looked at things like H2O2 content, malondialdehyde, MDA, content, and antioxidant enzyme activity. For genetic changes, they performed whole genome resequencing to identify SNPs and indels. And for methylation, they used whole genome bisulfite sequencing.
It sounds like a very thorough study. Were there any limitations or areas where further research is needed?
Of course, while the study provides valuable insights, the specific mechanisms governing the inheritance of abiotic stress responses across generations remain elusive. Further research is needed to fully understand the interplay between DNA damage repair, ROS signaling, and genomic methylation. Also, it would be interesting to see if these findings hold true for other plant species.
That's a good point. So, what's the big takeaway from this research?
The big takeaway is that spaceflight stress can have long-lasting effects on rice genetics, and these effects can be passed down through multiple generations. This is due to a complex interplay of oxidative stress, DNA damage, epigenetic modifications, and gene expression changes. Understanding these mechanisms is crucial for enabling plant cultivation in space and for improving plant resilience on Earth.
This has been incredibly informative. Thank you for sharing your expertise with us.
My pleasure. It's exciting to see how space research can contribute to our understanding of plant biology and potentially benefit agriculture.
Before we wrap up, could you elaborate a bit more on the role of alternative splicing in this process? It seems like a key piece of the puzzle.
Certainly. Alternative splicing, or AS, is a process where a single gene can produce multiple different mRNA transcripts, and thus different proteins. The study found that spaceflight stress significantly affected the splicing patterns of certain genes in rice, particularly those involved in stress response and ROS scavenging. This suggests that AS is a key mechanism for plants to regulate gene function under stress.
So, it's like the plant is fine-tuning its response to stress by creating different versions of the same protein?
Exactly. And the study also found that these changes in splicing patterns can be inherited across generations, further highlighting the long-term impact of spaceflight stress.
That's amazing. It really underscores the complexity of plant responses to environmental stress. One last question. Did the researchers identify any specific genes that seem to be particularly important in this process?
Yes, they did. For example, they found that genes involved in calcium ion signaling, such as calmodulin and calcium-dependent protein kinases, were upregulated in response to spaceflight stress. They also identified several transcription factors, like ERF, MYB, and BZIP, that seem to play a role in regulating gene expression under stress.
So, these genes could be potential targets for future research aimed at improving plant resilience?
Precisely. By manipulating the expression of these genes, we might be able to enhance plant adaptation to spaceflight and other stressful environments.
This has been a truly enlightening conversation. Thank you again for sharing your insights.
You're most welcome. It's been a pleasure discussing this fascinating research with you. Well, folks, that wraps up our discussion on space mutagenesis in rice. I hope you found it as interesting as we did. Until next time.